The start codon is the first codon of a messenger RNA transcript translated by a ribosome. The start codon always codes for methionine in eukaryotes and a modified met in prokaryotes. The most common start codon is AUG. The start codon is often preceded by a 5' untranslated region. In prokaryotes this includes the ribosome binding site. Alternative start codons. Alternative start codons are different from the standard AUG codon and are found in both prokaryotes and eukaryotes. Alternate start codons are still translated as met when they are at the start of a protein. This is because a separate transfer RNA is used for initiation. Eukaryotes. Alternate start codons are very rare in eukaryotic genomes. However, naturally occurring non-AUG start codons have been reported for some cellular mRNAs. 7 out of the 9 possible single nucleotide substitutions at the August start codon of dihydrofolate reductase were functional as translation start sites in mammalian cells. In addition to the canonical methionine met and AUG codon pathway, mammalian cells can initiate translation with leucine using a specific lysosyl tRNA that decodes the codon CUG. Mitochondrial genomes use alternate start codons more significantly. Prokaryotes, E. coli uses 83% AUG, 14% GUG, 3% UUG and 1 or 2 others. Bioinformatics programs usually allow for alternate start codons when searching for protein-coding genes. Well-known coding regions that do not have AUG initiation codons are those of LAC-I and LAC-A in the E. coli lac operon. Standard genetic code, A. The codon AUG both codes for methionine and serves as an initiation site. The first August in an mRNA's coding region is where translation into protein begins. See also, Central Dogma of Molecular Biology, Codon, Messenger RNA, Stop Codon, Transfer RNA, Translation, External Links, The Genetic Codes. Compiled by Andrzej Elsanowski and Jim Ostel, National Center for Biotechnology Information, Bethesda, Maryland, USA. 1. References, Lobanov, A.V. Churinov, A.A. Hatfield, D.L. Gladyshev, V. Dual functions of codons in the genetic code. Critical Reviews in Biochemistry and Molecular Biology 45, 257 €65 doi, 10.3109/104092310037869094. PMCA 3311535. PMIDA 20446809. Ivanov IP, Firth A.E., Michelle A.M., Atkins J.F., Baranov P.V. Identification of evolutionarily conserved non-AUG-initiated N-terminal extensions in human coding sequences. Nucleic Acids Research 39, 4220 €4234. doi, 10.1093 PMCA 3105428. PMIDA 21266472. Peabody, D.S. Translation Initiation at Non-AUG Triplets in Mammalian Cells. The Journal of Biological Chemistry 264, 5031 €5. PMIDA 2538469. Stark, S.R. Jiang, B. Pavan Eternad, M. Prasad, S. McCarthy, B. Pan, T. Shastri, N. Leucine tRNA initiates at CUG start codons for protein synthesis and presentation by MHC class I. Science 336, 1719 €23 doi, 10.1126/Science 1220270. PMIDA 22745432. Davia, T.E. Molecular Biology. A New Start for Protein Synthesis. Science 336, 1645 €6 doi, 10.1126/Science 1224439. PMIDA 22,745,408. Blattner, Fr. Plunkett G. Block, C.A. Penner, N.T. Burland, V. Riley, M. Kaleidovides, J. Glissner, J.D. Rode, C.K. Mayhew, G.F. Gregor, J. Davis, N.W. Kirkpatrick, H.A. Godin, M.A. Rose, D.J. Mao, B. Shaw, The Complete Genome Sequence of Escherichia coli K12. Science 277, 1453 1462. doi, 10.1126/science. 277-5331-1453. PMIDA 9278503. Sacerdote, C. Fiat, G. Dessen, P. Springer, M. Plumbridge, J. A. Granberg Monego, M. Blanquet, S. Sequence of a 1.26 kilobyte DNA fragment containing the structural gene for E. coli initiation factor IF3, presence of an AUU initiator codon. The EMBO Journal 1, 311 315. PMCA 553041. PMIDA 6325158. Misiakas, D. Jagopoulos, C. Reina, S. The Escherichia coli heat shock gene HTPY, mutational analysis, cloning, sequencing, and transcriptional regulation. Journal of Bacteriology 175, 2613 2624. PMCA 204563. PMIDA 8478327. E. coli lactosoperon with LAC-I, LAC-Z, LAC and LAC-A genes GenBank, J1636.1, Farabor, P.J. Sequence of the LAC-I gene. Nature 274, 765 769 doi, 10.1038/274765 AO. PMIDA 355891. NCBI sequence viewer v2.0, Nakamoto T. Evolution and the universality of the mechanism of initiation of protein synthesis. Gene 432, 1 6 doi, 10.1016/J gene. 2008.11.001. PMIDA 19,056,476 A.